Thank you so much for doing an interview with us. We're really excited. First and foremost, would you be able to tell us a bit about your background and what it took for you to become a scientist here at the University of Toronto?
Sure. So I started out, I wanted to be a medical physicist. So I started actually a physics engineering program in Norway, where I'm from, and graduated from that with a master's degree. But at the end of that, I became really interested in radiation biology as part of that kind of education. And so I started doing research in radiation biology, and then I did more and more biology, and so started a PhD there. I was really lucky. I got to go to McGill University in Montreal for about 6 months during my PhD to expand my horizons, learn about new things, and it was really informative, and maybe played a small part in bringing me back to Canada later in my career. So I graduated in Norway with a PhD, and then I did a postdoc in the Netherlands. And then I came to Canada in 2008, first as a scientific associate at Princess Margaret Cancer Centre. And then I got my own lab and became also a professor at the U of T.
Wow, so you traveled quite a bit then. Did you ever consider perhaps starting your lab not in Toronto or not in Canada?
I think by the time, I was at a place in my career where that was actually something I wanted and became possible, I was already in Canada and really loved being here, both in terms of being in Toronto, which I really loved in terms of kind of I like the size of it, the diversity, all the opportunities in research with many big institutions. Was new to me. I came from smaller environments, and I really enjoyed that. So I think by the time that I both wanted and could do that in my career, it was this was really where I wanted to be.
Oh, okay. I think a lot of students in MBP can really relate with wanting to become a medical physicist. I feel like a lot of students come in here thinking about that. And it's really exciting to see how your path kind of changes as you progress throughout your career.
Yeah, I think that is interesting, and sometimes, I also have, teenagers and they're very worried about choosing the right thing right away and taking a straight path. But I think sometimes those windy paths can give us maybe a different perspective on things so that we bring something unique, I think, to the table in research. And so, sometimes, although I think, I spent a lot of time trying to learn quantum mechanics or something that I don't use in my job now, I think that those experiences, still shape us, shape the way we think about things, the way we think about problems, and can actually be quite enriching for the research field, to have people with different backgrounds. So I don't think people should worry about that too much. They should just kind of pursue what they are interested in. And then I think we always find our home at the end of it.
Yeah, that's good advice. As a scientist, what does your group primarily work on? I know I'm more familiar, but for a lot of the incoming MBP students who may be interested in rotating, what are some of the projects that your lab is currently working on and areas of expertise?
Yes, I think all the problems that we work on, I could say that they're maybe rooted in problem of redox homeostasis in cancer, but in very different ways. So we're interested in how the changes in the redox balance, so the balance between oxidative molecules and reductive molecules in cancer cells, expose certain vulnerabilities that we could take advantage of in terms of new molecular targets. So right now we have a big focus on pancreatic cancer. We are very interested in some of the antioxidant molecules in pancreatic cancer that they're really dependent on for their survival and proliferation, and how we can target those, and especially then in combination with other treatments such as radiotherapy or immunotherapy. And so that is one focus. And I would say that this redox homeostasis is also very tightly coupled to the metabolism of the cells. So those are two areas of expertise in my lab. And we're also interested in redox homeostasis, I would say, coming from the fact that a lot of cancer cells have low levels of oxygen. And this is because of both high consumption of oxygen in tumors and because of sometimes poor oxygen delivery. And this is a problem in cancer because it makes cells really resistant both to therapy, and it also makes the cells much more aggressive. And so we have also research lines where we're really trying to better understand the biology of these hypoxic cells and what their particular vulnerabilities might be. And our current focus is actually on the role of oxygen in protein folding. And we're interested in that because we know a lot of those very aggressive phenotypes in cancer cells and in hypoxic cancer cells are driven by secreted proteins. And secreted proteins have to fold in the endoplasmic reticulum before they're secreted. We know that most of them use oxygen in that process. So it's an oxidative folding process. So we're trying to find out how cancer cells are able to fold these proteins in the absence of oxygen and still secrete all these molecules that confer aggressive behavior and bad prognosis for patients. And so the overall goal of that is that, when we understand that pathway, maybe we can target it and prevent all that secretion. So that is a more fundamental biology question that we think, on a more long-term scale could have big impact on how we think about targeting hypoxic cancer cells.
That's really interesting. Are there currently in clinic a lot of therapeutics that are targeted toward some of the signaling pathways that are affected by these redox reactions, for example?
There's a lot of focus right now on targeting metabolism. Those metabolic pathways support a lot of different things in the cells, but including redox homeostasis. And that's because some of the metabolites that cancer cells make a lot of, like NADPH, for example, is the molecule that really supports the reductive power of the cells and their ability to deal with reactive oxygen species that they make a lot of because of their high proliferation and their high rates of metabolism. So in a way, targeting these metabolic pathways, secondary to that is really, I think, disrupting redox homeostasis in cancer. And so these are largely, experimental at this point, but really receiving a lot of attention, trying to disrupt nutrient uptake or metabolism of those nutrients more downstream.
Yeah, when you mentioned metabolism work, the area of metabolism has blown up a lot recently. And so how do you think the tumor microenvironment, metabolism, and metabolomics in general affect protein folding?
Yeah, that's a really, really interesting question, I think, and something that we don't know a lot about. So we know that there are certain metabolites that are really important for protein folding. And so one example is ATP, for example, that chaperone proteins are dependent on ATP to function. But we think that ATP is usually available in cells and in cancer cells. They're very good at maintaining their ATP levels. That rarely becomes limiting for the folding. There's other metabolites like FAD, for example, that's used for electron transfer by certain proteins. And so that is an interesting area of research, and we're actually looking into this. So we're quite interested in how, for example, metabolites that may be made other places in the cells are transported to the endoplasmic reticulum so that they can be used in protein folding. But this is very experimental, and this is something that's one of our research interests. We know in the tumor microenvironment, there's other parameters that really affect protein folding, such as pH, for example. So in the tumor microenvironment, often the pH is low, and we know that affects activity of some folding enzymes. That's also interesting. And then, of course, oxygen. If you open any textbook in molecular biology about oxidative protein folding, you'll see that oxygen plays an extremely important role, although now we know that cells can do this without oxygen. And so that's why we're trying to find these oxygen-independent pathways.
Yeah, that's very important. I think especially for just foundational knowledge, for like my own work, where I look at the combination of immunotherapy and radiation therapy. So very important work that you're doing. Has your understanding in protein folding helped? Do you think that it's helped guide current cancer therapies? And what do you think is the future for this current field?
Well, I think there hasn't been a lot of focus on this yet in cancer therapy. So there has been some focus on other more sort of cytosolic chaperone systems, for example, for cancer therapy, targeting the main chaperones like HSP90, for example, where there's been some exploration, but those are very general pathways. We think it's important to understand more specific pathways that maybe support the folding of a subset of proteins that are particularly important for cancer cells, because that's going to give you a bigger therapeutic ratio eventually. I think it's interesting, because if we kind of think of the dogma of molecular biology, right? We now have agents that target gene expression and specific gene expression on every level, right? We have drugs that affect the epigenome. We have drugs that affect the transcription in hypoxia, for example. We have drugs that target protein degradation. But we don't really have much all the way downstream to protein folding. So I do think that that's an untapped potential, and that's why we're focusing on it, because if there are common pathways that support folding in certain situations, like hypoxia, that only cancer cells are dependent upon, that would potentially give us a therapeutic ratio when we target that. So stay tuned.
Thank you for answering all of our science-related questions. We're going to switch gears a bit. You've been a PI for quite some time now, so you've seen a lot of graduate students come and go, and you've seen a lot of growth in graduate students in general. And so when you're looking for potential graduate students, what are the kind of qualities that you look for?
I think actually the most important quality I look for is what you just mentioned, is actually desire to learn and then desire to grow. Because, I think it's really important to remember that graduate studies are that. They're studies. they are education in research. They're education to become a better researcher or scientist. And so I think that is really important. We shouldn't think of it as, if you do a master's degree, we shouldn't think of it as a 2-year exam and then you pass your exam at the end of the 2 years. It's really about what you learn, during those 2 years that you benefit from later. So that is something, it really motivates me when I feel like students, really want to learn. That helps me a lot. And it's probably one of the most important qualities I look for. We are really lucky to be at a top university. We have fantastic students. We get a lot of students that have, they have excellent grades. Everybody who gets into our program essentially, right, have very strong academic background. They're hard workers. They're very motivated. So we're really lucky with that. But I think at the end of the day, it's really this sort of desire to be a better researcher, right? Try to absorb as much as you can. The other thing I really look for too, actually, is someone who's going to be a good colleague for people in my lab. I think graduate student studies are always hard. If you haven't struggled, it's not a graduate degree, right? Which is hard to hear sometimes, but I think that's true. It's really part of it. And it's so important to have a good environment in the lab, and people can be supportive of each other. They can learn from each other. They can receive mentorship. They can give mentorship. I pay a lot of attention to that as well, that I think somebody will be a positive contributor to the environment. In addition to that, there's, of course, sort of regular criteria. Do you have sort of background in this field? Do you have previous lab experience? And I think, something like previous lab experience and accomplishments are nice. It's a bonus. But I actually think that most students, if you're smart and hardworking and dedicated, you can learn techniques. I don't think that's the most important thing. A lot of students focus a lot on it. Actually, when they apply for jobs, they talk a lot about that they've done qPCR and Western and DNA isolation, which is great. Gives you, okay, so you don't have to learn that. But at the end of the day, I think it's sort of the other qualities, your own drive and passion and interest that contribute to your success more than the techniques you already know when you start in the lab in graduate school.
Yeah, I think you really put it in such a succinct way. Grad school is so difficult, and I don't think people really understand how hard it can be. And so it's really important that I think people are passionate and enthusiastic about your work, because when nothing works, you need something else to push you and motivate you. That being said, though, you also touched on mentorship, which I think is extremely important in helping support and guide these students who kind of don't know what direction they should go into next. And so what is your specific style of mentorship?
Yeah, that's an interesting question. I think what I've learned over the years about mentorship is that different people need very different things. I think that's the most important thing I've learned. So there's really no, in my mind at least, that I've found a one size fits all when it comes to mentorship. So I try to identify for each student what are their strengths that they should build on and what are weaknesses that they should work on to get better at. But I think the approach I take to that differs a little bit when it comes to the student, and it could also differ in time, because everybody, in their graduate journey go through different times. There's times where it's really hard and nothing works, and all you need is really support to get you through it. And then there's other times, where there's more room for being pushed and challenged. And so this is a really hard task, I think, for us as supervisors. It's something that we are never trained in. All we do is we take our own experiences from our mentors, and then we gain some experience, over time when we have the privilege to have students in our lab, but it's something that we have to kind of work our way through and sometimes try and fail a little bit at as well. So anyway, that's really my approach. I try to find what everybody needs and at different times and try to help with that as much as I can. Whether that's successful or not, I don't know.
I think that's a very good way to define mentorship, the balance, and a very fine balance between pushing and challenging your students, but also making sure that they have the support they need when they fall. What are the things that you kind of learned over the past years on how you can make sure that your students are succeeding when they're moving forward, but also ensure that they do have the support that they need?
I think it's very important to kind of leave grad school with this feeling of accomplishments, in the sense that this was hard, but I did it. And sure, you had help, right? You had your colleagues in the lab, and you had your supervisor, and you had your committee and other people give you support. But at the end of the day, this was something you did and that you should be really proud of. I hope that the students graduate with that, and that gives them sense of confidence, accomplishment, and that they are able to overcome things that are difficult. I think that's a really powerful thing to know, because there's going to be lots of hard things coming up after grad school. And I hope at least that you have that experience, that it was really hard, but you overcame it and you did it. Of course, I also hope that the purpose of all this is really to become better researchers and better scientists, and that they feel that they have a certain skill level at that they can ask good questions, they can decide about good approaches to attack that question. They can evaluate data in a critical but also constructive way and present research going forward. So all of those things, I hope people take with them. And I think all of those skills is something that everybody would benefit from, no matter where they went after grad school. Previous students from my lab have a wide variety of jobs now. And when I look at those jobs, I think that they can use what they learned no matter what.
I really agree with you. I think that oftentimes it's the most difficult things that are the most rewarding. And I'm not there yet, but I can especially imagine how amazing it must feel to achieve a PhD or even achieve a master's degree after, running that same Western blot 20 bajillion times and hoping that this one time it works. So I think that's a really good piece of advice. That being said, though, your lab also seems like a very great environment to foster scientific curiosity and a lot of scientific inquiry and growth. And so for incoming rotation students, what's the kind of style of rotation that they would typically expect to have when they come to your lab?
The rotations are short, so to get the most out of it, I think it's most useful to be paired up with someone who has a project that's running and there's some techniques that are working And so you can really get a flavor of what the day-to-day would be in that kind of project. And we also then get a good impression of how the student would thrive in this environment. So I do try to give students a small piece of the puzzle, a small independent piece, but that is very tightly linked to something else that's going on with someone who's experienced in the lab that would serve as a good mentor and both be able to help that student, but also help evaluate whether that student, is a good fit for our lab.
Yeah, I think rotations are so short, and I think they're super important in identifying, what's a good space for you and what you may like. Do you find a lot of rotation students typically enjoy working or like shadowing? Like, do they get a lot of hands-on time when they're doing these experiments? Or do you find they typically kind of want more of a balanced approach where they have some time where they're doing desk work and reading and learning about what the lab does, and then going to lab sometimes to do experiments?
Yeah, we try to do both because it's obviously really important that people, understand well the background for the work they're doing. So we try to achieve a balance. At the end of their rotation, they can present their work to the lab. And it's not that we have enormous expectations of what can be achieved in a short period of time, but at least we can hear from that student about how they see the project, the research question, and what they were able to do during their time. So yeah, we do try to get a balance about that. It's important to work in the lab and get a flavor for what that's like and for us to see how the student adapts to that. But just as important really to find out, if you're really interested in this research question at the end of the day, that's the most important thing. Is this a research question you can really be passionate about and really want to immerse yourself in? That's, I think, the most important thing about the rotation, to find out.
Yeah, I definitely agree. Well, we both also touched on how difficult graduate school can be numerous times already. When you were going through grad school, what were the kinds of things that helped keep you motivated during your difficult times?
Yeah, I think people are very different there. I think, I was I still am, I think, very passionate about the problem. I just really want to figure it out. And that sort of would keep me going. Letting it go was just not an option. But I think something that helped me was this community in labs where if maybe my project wasn't going great, somebody else's project was going great. And so I think that is something I've also learned, that it's really nice if people in the lab, all have a small part in each other's projects, because it can really help you through those hard times. And it's so much more fun if we think that, Cindy's success is also a little bit my success, right? Because we collaborate a little bit on this piece, or I help out a little bit with this, and I know her project really well. And so I think having this community where we all have these independent projects and our goals, but we also feel like there's a bigger picture and that all these research questions are connected, and we can take some pride and success in what else is going on in the lab, I think is quite helpful. The other thing that helped me is I had a mentor, a supervisor that was just sort of the incarnation of positive. I literally remember going into his office and thinking that I'm never going to finish and nothing works. And half an hour later, going out of his office thinking that we'll probably win a Nobel Prize. And, the ability of a supervisor to turn that around, right, and help you see the sometimes bigger picture, but sometimes also sort of alternatives and ways to get at something that you hadn't seen that you could take advantage of, was really, really helpful for me. And I try to sometimes be like that because it really helped me. But I think that's amazing, actually, when I think back on that, on just his sort of just genuine sort of positive attitude and ability to make other people feel like that was really valuable too.
Yeah, I think you really hit it on the nail. I think the power of, this sounds kind of cheesy, but the power of teamwork, and with teamwork comes with diversity of thought, and all that fosters and establishes a sense of community that will like uplift you and, empower you to do all these very challenging and difficult things. And so what are the things that you do in your spare time for fun that aren't science?
Yeah, I do. I do a few things. I like doing sports. I really like skiing. I'm Norwegian, as I mentioned, and that's kind of our favorite pastimes. And also, when I came to Canada after a few years, I thought I should assimilate. So I started playing hockey, which I'm terrible at. But I play in a really sort of a low level old ladies rec league, which is a ton of fun. During the pandemic, I had to find, something new to do because none of these things were really possible. And it was kind of funny because I took up sewing. So I started making quilts and bedspreads. And I think it was, I actually think it was some kind of need in us to create something, when we're stuck inside and we just feel like we're not doing anything. It was liberating to take up something, to make something useful. I'm also terrible at that, but I do find that actually a little bit liberating in a way because, I feel like in our jobs as scientists, we're really focused on always, right? Ambition and excellence and perfection. And if you talk to any of my students, they'll tell you how much I pester them about, figures looking perfect and aligned and beautiful. And, so both content and presentation, right, is really high bars and standards for. And so I actually find it liberating in my time off science to do something where the bar is really low and I'm bad at it. And I do it and I look and I think, oh, that looks terrible. Oh, well, it'll keep me warm, And in a way, that's been quite fun to just go with the flow and have fun with it, I guess.
Yeah, I think scientists or even graduate students or just academics in general are all very high achieving people. And I think we're all to varying degrees a perfectionist. And so I think it's super important that you have something that's just like, you will be terrible at this and you will suck at this, but it's purely for fun. I have started like longboarding and I am absolutely horrible. I also think it's very impressive that you are, this active while maintaining like a super high functioning lab. Very, very impressive. So hopefully one day.
Well, I think sometimes it makes us better. One of the things I really enjoyed actually about hockey was that, it's an hour where I don't think of anything else but the game. That is also liberating because scientists and mother all the time, at other times I think about either science or what's for dinner or this kid needs to be picked up and brought there. And, all these things are happening at the same time always. And doing something like an intense sport just kind of lets you, let go and not think about anything like that for an hour. And it's actually really, I think, refreshing and important for the wellness for sure.
Yeah, 100%. I think it's like a brain break or something.
Yeah.
During these moments, do you ever sit back and think about what would your job be if you didn't choose to pursue science?
Yeah, that's a great question. It's hard. I've been in it for so long. I don't know. I do like writing, which is actually something that I think have come into good use in science. But I do like writing. Used to at least be decent at it. So I don't know. Maybe I'd be some kind of writer, a journalist, or something like that if I wasn't going to do science. That's a lot about communication, and which is super important to be successful in science as well. But that's something I liked, I guess, that I didn't pursue. But I think for me, it was always natural sciences that I was really interested in. And I think, I could have stayed on my path to be a medical physicist, and I think I would have enjoyed that too. But I kind of do think I would have always been drawn towards doing research in that field, not sort of work in the clinic, but be drawn to development and, in maybe in more the engineering space than in the basic science space.
Yeah, I think in any field, especially when you're in the clinic, there's always overlap between new research and, new therapeutics. And you're always like kind of drawn, or at least I feel like academics are always drawn to the what ifs and how can I improve this? And all these other things that kind of build upon each other that just bring you towards research. My final question for you, which I think you briefly mentioned because I didn't know that you played hockey, very Canadian, is what is a special skill or talent that you have that most people don't know about?
Oh, gosh, yeah, because I just told you about all the things I'm terrible at. I don't know. I think maybe one interesting thing that people or many wouldn't know about me is that I have another job. In addition to be scientist, I serve as a consul general of Norway in Ontario, which means that I'm the representative of the Norwegian government, which is really interesting and something that I was offered some years ago and was very surprising to me, but really exciting. So I accepted to do that, next to my scientist job, and it's something that's been really enriching just to do something else, meet a lot of people that I wouldn't have otherwise met, and learn a lot about topics that I wouldn't have otherwise learned about. So bilateral relationships and different exchange of knowledge and approaches between the countries has been really interesting. So that's probably a pretty unique combination of scientist and consul general, I would think.
Yeah, it's very unique. I don't think there are many people like you in that sense. And it must be very difficult to balance too, being a mother, being a scientist, being consul general, being this active, like a lot of stuff going on.
Yeah, I probably didn't need an extra job, I'll admit that. But it's one of those things that, when it comes your way, it's such a unique opportunity that you just feel like can't turn it down, right?
That's true. Well, those are the questions I prepared today. Thank you so much for joining us for our interview.
Thanks, Cindy, for having me. It's been a pleasure to talk to you, and best of luck to you in your graduate studies. And good luck to all the incoming MBP students as well, as they're really, entering a super exciting time of their lives. So best of luck. Sure.
Thank you so much. Bye.
Thanks for having me.